So the title of today's lecture is Proteins Found in Blood. And we're going to spend some time emphasizing a few of these, but I'm going to break sort of in the middle to really kind of talk about proteins in general, because it may have been a while since we've discussed proteins or you've talked about them in any of your classes. So let's start number one. The most prominent protein found, and these are found in plasma. Now you're going to find proteins in the cells, of course, because all cells have lots and lots of proteins in them. And red blood cells are covered in hemoglobin and all kinds of stuff. But like just specifically plasma. Remember plasma back in our first lecture. Plasma proteins make up about 7% of plasma. And so these are the things that make up that 7%. First one is albumins. These are proteins that pretty much their whole job is to help maintain a proper osmotic pressure. And I'll explain what that means. They make about 55% of all the proteins in your plasma. And like I said, they maintain osmotic pressure. And in this case, when we're talking about very, very specifically the osmotic pressure related to plasma and cells and proteins and things like that, it actually gets its own special name. It's called oncotic pressure. Maintains oncotic pressure. Now, what that means is this. Inside of each cell, there are a lot of proteins by themselves dissolved in solution, okay? If we don't do the same thing in the bloodstream, what's going to happen is water will naturally get drawn out of the bloodstream into the cells at a really high rate. Now, it's supposed to go there anyway. Our cells need water and stuff, but it would go there at an even higher rate because water will have a tendency to move through a membrane in the direction where there is a higher concentration of solutes, like albumins and proteins. So having a high concentration in the bloodstream basically kind of levels the playing field, making sure that we have an even distribution inside the cells and inside the bloodstream so things don't go too far one way or the other. So this really is important to try to maintain a fluid balance between inside and outside of the cell. The next type of protein is called globulins. And these proteins they make up about 38%. So these two together make up a majority of these. These are responsible for immune response because these are antibodies. So all antibodies are globulin. Okay? So that's what their job is basically to fight infection, fight disease, help to mark and target things that don't belong in our bloodstream so our white blood cells can effectively attack them, we can remove them. The next one that makes up the third largest component is something called fibrinogen. Fibrinogen makes up about 7%, ironically, of the 7%. And this helps with clotting. These guys are kind of on standby, waiting for a cut or fluid loss, and they'll then be converted from fibrinogen into a spider web like material called fibrin to help kind of catch red blood cells and start forming clots and stuff. But in this form, they really don't clot up like that. So the next one.
I need a custodian to please come to the attendance window. A custodian to the attendance window.
As you can tell by my recording, we need a custodian at the attendance window. I'm recording this after school, and so you're going to get some of the sounds thereof. So just bear with me. Number 4 is regulatory proteins. Regulatory proteins. Okay. These make up less than 1%. They help with gene expression. Basically, whether or not a gene is switched on or off has everything to do with these. That's supposed to be a less than sign. Has to do with these regulatory proteins. Next, number 5, clotting factors. Now, I mentioned that fibrinogen is a big part of that. But there are other clotting factors besides fibrinogen, and they make up also less than 1%. Their job, of course, is to help convert fibrinogen into fibrin, which is that spiderweb-like stuff. Okay. So those are the 5 main proteins that you'll find in blood plasma. Now let's talk just for a second about proteins. Proteins are amazing. Okay. I'm going to mess this up here for you. The sweet fruit of the DNA tree. You're like, what? They're fruits? No, they're not fruits, but like in biology, I would go so far as to say proteins are probably the most amazing substance that exists on the earth. Yep, I said it. They can be so diverse in function. Some of them, you know, helping to clot blood. Some of them maintaining osmotic balance. Some of them being right there on the screen. Your fingernails are a type of protein. The same type of protein as your hair. It's called keratin. Proteins that are enzymes that catalyze reactions. There's tons and tons of different types of proteins, which you're going to look at here in just a little bit. Okay, structure of a protein. Structure of a protein. They are chains of amino acids the sequence of amino acids sequence, and if you don't remember, sequence just means order. Whatever order the amino acids determines what the protein is. That's it in a nutshell, folks. That's the difference between the keratin in my fingernails and the enzyme pepsin in my stomach that's breaking down proteins from my lunch. The difference between those two is the order or the sequence that the amino acids go in. So let's look at an amino acid here for a second. And I'm going to try to break it down. I've got some colors to use, so let me try that. All right, so on this side is the amine group. Okay, so I've got N, H. That's not a very good H. H. Okay. Then in the center, I've got my regular stuff here. Oh, let me go back to blue. Sorry. Black. All right. Then I've got a carbon there with a hydrogen there. And then on the other side, I have this carboxylic acid group, C, double bonded oxygen, O, OH. Then down here below, I'm going to use some blue, R. So I'll circle that one. Circle my amine group with my weird hydrogen there. And circle that carboxylic acid group. Okay, so NH2 is considered the amine group. And it's called that because it's NH2. Amine, very similar to ammonia. Ammonia, which would be NH3, or ammonium ion would be NH4. Then we've got the COOH on this side, which is the carboxylic acid group. Now you can quickly see why this is called an amino acid. An amino acid because it's got an amine group on one side and a carboxylic acid group on the other side. This wants to get rid of hydrogen ions, therefore it's an acid. Now, you looked at here and you're thinking, whoa, that's weird. I don't remember an element with R. That's because there are none. I'm just kidding. Okay, that's the R group. R group means that we can swap this one out for pretty much just tons of different things. And every amino acid has all of this in common. Like, all of this is pretty much identical in all amino acids. Some are right-handed, some are left-handed, like you'll have the R group here as opposed to here. But other than that, this whole thing is going to be the same. It's the R group that's different. So let me give you some examples here. So we've got an amino acid, and I won't use the different colors on this one. Amino acid right here. Well, the simplest one we could make would just to take that R group and swap it out for an H right there. And if we do that, we have an amino acid known as glycine. That's glycine. That's its structure, where the R group is an H. Now, if we take that and swap that out for an R group that is a methyl group, or a CH3 here, or like methane, which is CH4, we take NH, H, C, C, O, H right there, O, OH, and down here we put one of these in, and this guy is called alanine. And again, what makes it different is what's down here in the R group. Now, amino acids will be formed together in a reaction called a dehydration reaction. So this all happens during a process known as protein synthesis. If you remember back to biology, you have the DNA in the nucleus, and then you make a piece of RNA that goes out to the cytoplasm. It meets up with a ribosome. The ribosome basically reads it and then attaches these tRNA molecules that are carrying amino acids, and then starts assembling them together. Well, part of the assembly process is to take this H and this OH, put them together, pull it out as water, like that, and that's called a, oh, that's not how you spell it, dehydration reaction. And then when you do that, you end up with a molecule that looks like this. And C, C, double bonded oxygen, and C, C, O, O, H. And it'll look very similar to what we've got above, except these two are now stuck together with a covalent bond. Oh, we've got a hydrogen there too. H. Okay. And this covalent bond that is now in between those two, because the word we use for proteins and amino acids is peptide, this is called a peptide bond. Now, this thing, because it's two amino acids joined together, is now called a dipeptide. And when we join three or more together, it's called a polypeptide. Now, what happens after that is, because of all these different R groups and all the ways that they're going to react to one another. Now, I've only shown you 2 amino acids. There's at least 20, and I don't remember off the top of my head anymore. I'm thinking like somewhere like 36 or something like that. There's 20 we have to have in our diets because our body can't manufacture those. They're called essential amino acids, but like I think there's like 36 or 40 different ones, and they can be in any order. But when our body makes proteins, or living things make proteins, they assemble them in a very specific order to be able to do a job. And if they're not in that order, they can't do the job. Because what happens is, because of the way they're put together, the order, the sequence they're put in, they fold up. And they create these four structures, these four structural levels of proteins. Now the first one we just talked about, okay, and that is number one, is called the primary structure, which is nothing more than the sequence of amino acids. After the primary structure, we have the secondary structure. Because this long polypeptide chain, like I said, when you have 3 or more, and a lot of times they're thousands and thousands of amino acids long. Some of them like tens of thousands. I think one of them is like 33,000. That's the protein Titan. It's also the longest word in the world, because a protein's real name is the name of all of its amino acids together, because this would be like glycylalanine, you know, if it were just that dipeptide, but then multiply that times 33 names like that all stuck together. What happens when you get these big chains together is they start folding up, because these R groups start reacting with one another and they start folding. That initial folding, initial in this case just means like first folding of the, and I'll abbreviate polypeptide as PP chain. Yeah. Polypeptide chain. It starts folding into something called these alpha helices, which look like these little squiggles or swirls like that, and these things called pleated beta sheets, where they start kind of making these back and forth like sheets, and one folds upward like that, and then they fold down in the middle and up like that. You don't have to know this for the test, I'll just kind of give you an idea. So that initial folding. And then it folds in different ways, and that's called the tertiary structure. As the final folding of the polypeptide chain. Okay? And that's the tertiary structure. You're like, well, how can that be final if there are 4 structural levels? Here's the reason. If you end up with this last structure, is called the quaternary structure. Is where you have 2 or more polypeptide chains that form together. There's an important concept I want to emphasize here, and that's this. How proteins fold determines their function and what they do. The trouble with that is this. They're very sensitive. Proteins are very sensitive to 2 things. Okay? If they unfold, they don't work. And that unfolding, the name for that is denature. If proteins denature, they no longer function. 2 things that would make them denature very easily. Okay? They denature if they're in the wrong temperature, which is why having a high fever kills people. If you get it too high, it denatures your enzymes in your cells that carry out and catalyze your reactions. and therefore your nerves and your muscles and everything else fails, and the cells die and doesn't work. Okay. The other thing is pH. Your enzymes are sensitive to pH. They have to have a very specific pH that they work in. They have to have a very specific temperature they work in. They get outside of that range and they don't function, or they don't function correctly. Okay, beyond that, let me give you a few categories of proteins in general, and then we'll call it quits for today. Okay, so categories of proteins. Okay, first one is enzymes. Probably my favorite one because they're the most diverse. I don't even know if that's true. They have the most array of functions. They're catalysts. They basically make reactions happen. Some that like build stuff up, some tear stuff down. So like there's an enzyme in your cells called DNA polymerase, and basically it makes new DNA molecules for replication, so it's building stuff. There's another enzyme that I mentioned earlier called pepsin in your stomach, and it's breaking down connective tissue and things like that, so it's tearing things apart. Either way, it's catalyzing a reaction, whether it's building things up or tearing things down. It's a catalyst. The next one is called a structural protein. And I alluded to these earlier, like keratin in your hair and fingernails. Okay. Or collagen, which is in your bone and cartilage. Bone and cartilage. I think it's cartilage, I think. Check me on that. All right, next one would be like a signaling protein. Signaling protein. And these would be like your hormones that basically tell other cells what to do or how to react. Regulatory, like I mentioned earlier, which are in charge of gene expression. Think about your DNA as like a switch panel, basically like a breaker panel in your house. Like some genes are switched on and some are switched off. So in some cells these genes are all switched off, and in other cells these genes are switched on. And that really is what largely controls what that cell does for a living, whether it's a nerve cell or it's a muscle cell or whatever it is. And ultimately it's regulatory proteins that will help determine that turning on or switching off, those types of things. Then we have transport proteins. Transport proteins carry stuff or help blood, which carries stuff. Hemoglobin's a good example because it carries oxygen. And then albumin is another example because albumin helps blood, which carries stuff, and albumin helps to regulate that osmotic or onconic pressure. So next one is sensory. These proteins will help us see and smell and stuff. If you smell cinnamon, that's because there is a protein in your nasal passages that reacts to that molecule and tells your brain, hey, that's cinnamon. Motor proteins, these seriously move, okay? Myosin is a good example. Myosin. And then basically these are ones that help move. There's motor proteins in our cells that actually help move things along as well, like vacuoles. Then we have defense proteins. Defense proteins would be like antibodies, globulin proteins, things like that, are defense proteins. And finally, storage. Storage proteins for energy storage. I thought energy was fat and energy was carbohydrates. Yes, that's true too. Okay. In egg whites, there is something called ovoalbumin, which is meant for the embryo to use for nourishment. In milk, there's a protein called casein. Oh, I misspelled it. My bad. Casein. It's a weird one. And that one is for energy for the developing child and infant and stuff like that, to be able to make energy. One that's got a lot of bad press lately is gluten. Gluten is a wheat protein. It is in the seed of a wheat plant, which breaks down to make flour and stuff like that. Originally, it's put in that seed to provide nourishment for the developing plant embryo, but some people are allergic to it, so it gives them trouble. So here's your categories, gang. Those are, looks like 9 different categories of proteins that exist in living things. So that's basically it. So proteins in blood, proteins in general, and categories of protein. Hope this helps.